Good morning, dear students. Today we are going to start another chapter, that is bacterial recombination. First point we need to understand is nucleoid. What is nucleoid? I think, or I don't know whether you remember it or not. We have learned this term in the beginning of this semester itself in the first chapter, Introduction to Cell and Molecular Biology, nucleoid, regarding the different types of cells, prokaryotes and eukaryotes. In that chapter, we have already covered this term. What is nucleoid? I'm just brushing up. It is the position occupied by single circular double-stranded molecule of DNA in prokaryotes. That is nucleoid. The position where the single circular double-stranded molecule of DNA occupies in the prokaryotes. That position is nucleoid. And what is plasmid? This term also we have learned. Plasmid, in addition to that DNA, double-stranded circular DNA that present inside the prokaryotes, it possesses an extra chromosomal structure. That structure is plasmid. What is its significance? Some significant important genes are located in this extra chromosomal structure, and that genes give the bacteria antibiotic resistance. Because of the presence of specific genes in the plasmid, that bacteria become resistant to certain antibiotics. That is the significance. Then what is episome? That also we have learned in that same chapter. Episomes means they are certain types of plasmid, but they can replicate freely and also can integrate into the bacterial chromosomes. And what is recombination? This term we have just finished in the former chapter, that is cell cycle. What is recombination? What is its significance? That is responsible to create variation in the next generation when compared to their parents, right? So, recombination, how it is happening in the case of prokaryotes, that is what we are going to learn in this chapter. As this bacteria cell is haploid, it is not possible much to undergo again division to divide the already had genetic material. It's not possible because it is haploid. But they are also undertaking this recombination process. And how that recombination takes place in the case of prokaryotes, that we are going to learn under different heads. They are, or different methods. They are transformation, conjugation and transduction. These are the different process of recombination that is found in bacteria. First, we can learn about transformation. What is transformation? It is the uptake of free naked DNA molecules from one bacterium to another bacterium. And the bacterium that is donating the DNA molecule is called a donor, and the one that is receiving is called a recipient. And who gave the evidence for this process for the first time? That is Frederick Griffith in 1928. In which bacteria? In pneumonia causing bacterium, Streptococcus pneumonia. What he found in that bacterium? As the rough strain bacteria are cultured along with the heat killed smooth strain bacteria. This rough strain get transformed into smooth strain somehow. And the reason behind that change means the change that convert rough strain to smooth strain is transformation. Okay, then I am going to explain to you what is transformation very simply. Suppose this is a bacterium, it is having its own single circular double-stranded DNA. When this bacteria is destroyed, its genetic material also get fragmented, and some of the fragment happen to get attached with the body of some other bacterial cell. and later then they are taken to this bacteria cells inside, and after that it get incorporated into its genetic material, its double stranded single circular DNA, and because of that incorporation, that genetic material become a recombinant one, and it gains some new characteristics. This is simply the transformation. And this transformation in bacteria takes place through three stages, which are the competence, binding, and uptake, that we are going to see next. Then what is competence? Competence means the ability of the bacterial cell to pick up a donor DNA fragment from its surroundings. If we consider a group of bacteria, in that group, maybe the whole bacteria are not able to pick up the donor DNA fragment. Only a few are capable to do that. And those bacteria cells that is capable to pick up the DNA fragment are called competent bacterial cell. How they become competent? Because of the presence of certain DNA receptor proteins on the surface of its cell wall. And what actually the competence, what is that process? It involves activation or formation of these receptors on the bacterial cell wall. And the number of these receptors vary from species to species. Okay, that is competence. Then binding and uptake. What it means? The direct interaction of the donor DNA fragment between the cell wall of the receptor. That is that process. And after that interaction, the donor DNA is taken into the recipient cell. Then next, we need to understand that what is the difference in the method of uptake in the case of gram-negative and gram-positive bacteria. In the case of gram-negative bacteria, for example, Haemophilus influenza, the donor DNA is taken up in its double-stranded form itself, in the case of gram-negative bacteria. But in the case of gram-positive bacteria, what is happening? The double-stranded DNA is first broken into fragments with the help of some enzyme called endonuclease. Means these enzymes act the bond that is present within the DNA, inside the DNA, inner bonds, and that is cleaved by this type of enzymes, endonuclease. And after making the circular DNA into fragments, then each fragment is acted upon by exonuclease. Exonuclease means this enzyme can act upon the bond that holding the nucleotides that is found in the extreme ends. So that enzymes are called exonuclease. And what they are doing? They convert the double-stranded DNA fragment to single-stranded DNA, and later it is taken up by that bacteria. That is the difference in the case of gram-negative and gram-positive. Difference in the process of uptake of donor DNA fragment. Okay, next we are going to understand about another process, that is integration. How the uptaken DNA fragment is incorporating to the DNA or the genetic material of the recipient bacteria. That is we are going to see now. I think I have seen that. Once more I will show you. The donor DNA fragment, it comes and binds with the receptor that is present on the outside in the cell wall surface, and as it binds there, the recipient bacterial cell invaginate to form some vesicle, or in some other case with the help of some other proteins, it taken up the DNA fragment inside the cell. And if it is in double stranded form, then convert it to a single stranded form. Then we know that this bacteria also possess its own double stranded circular DNA or its genetic material. Next, this DNA fragment is going, and first a cleavage happened here on one strand of this double stranded DNA, and that incorporated single strand get incorporated or integrated to this genetic material. Okay. Then after this incorporation, in the next round of division of this bacterial cell, then what happened? This strand, this incorporated strand acts as the template for the creation of its complementary strand in this region. And what will be resulted? The result is the newly formed bacteria. I will show you the newly formed bacteria. This is the newly formed bacteria, and its genetic material will be like this. That means that incorporated single strand become an integral part of this bacteria in the next round of its division. Okay, that is the recombination method that happens in bacteria by the process of transformation. Okay. Next recombination process found in bacteria is conjugation, and it is discovered by Lederberg and Tatum in 1946. What is conjugation? It is the one-way transfer of DNA from a donor cell to a recipient cell through a specialized intercellular between two cells intercellular connection. What is the name of that specialized structure? It is called the conjugation tube. So in a simple way, what is conjugation? It is the one-way transfer of DNA from donor cell to recipient cell through a conjugation tube. And the donor will be a male. Why it is male? Because it possesses an extra chromosomal plasmid. That extra chromosomal plasmid is called a sex factor or fertility factor, or otherwise called a F factor. And the bacteria that is possessing this factor is male, and the other one that lacks this extra chromosomal plasmid is female. Okay, then what is this F factor? This F factor also is a circular double-stranded DNA stay apart from the main bacterial chromosome. It is with a molecular weight of 35 into 10 raised to 6 Dalton and having 1 lakh base pairs. And the cell that possesses the F factor is called F plus strain, and the other one that lacks that F factor is called the F minus strain. And conjugation always takes place between F plus strain and F minus strain. Next, we are going to see that process. This is a bacteria cell. This bacteria cell possesses that double-stranded bacterial chromosome. Along with that, an additional extra chromosomal DNA is called the F factor. Then came another bacterial cell. In this, we can find that this cell is devoid of that F factor. So, this is female and this one is male. Or otherwise, this is F plus strain and this is F minus strain. Okay, then this F plus strain start to develop a tube-like structure. First, it's called F pilus. Again, I will show you a tube-like structure called first called F pilus, and as it get connection with another bacterial cell that is F minus, then that connection is called conjugation tube. Okay. Then after forming that conjugation tube, what next is happening? A cut is happening in one of the two strands of that F factor. Okay. And the cut end that is having 5 dash end. That is having 5 dash end. Suppose this is the 5 dash n and other will be the 3 dash n. That 5 dash n will directed through the conjugation tube towards the F minus strain. Okay. And as it completely transferred to this F minus strain, what will happen next? After the transfer of that F factors one strand completely to the other bacterial cell, what happened? That single strand acts as the template for the formation of other strand. And here also the strand that left here in this cell acts as the template for the formation of other strand. So in both cell, a double stranded sex factor F factor is formed. By that way, the two bacterial cell now become F plus strain. This also F plus and this also F plus strain having the F pilus. F pilus. F pilus. Both these bacterial cells are now having the F pilus to get attached with the other F minus strain. F minus strain bacterial cell to begin another round of conjugation. And one more term you need to familiarize along with that, that is rolling circle mechanism of replication. Rolling circle mechanism of replication. What is it? Rolling circle mechanism. It is the unidirectional. Means in one direction only the replication takes place. That type of replication is rolling circle mechanism of replication. Here we have seen that a cut or incision is happen in one of the strand, and that a single strand is moving to the other bacterial cell, and that acts as a template for the generation of the other strand, and that other strand is replicated by the way of this rolling circle mechanism. That means that the other strand is formed from the existing one in one direction, unidirectionally. That type of replication is called the rolling circle mechanism of replication. Okay, is it clear? Then I am going to explain some more points relating with this conjugation. That is, the F factor can exist in two different states. In majority of the case, that F factor will show that, suppose this is the bacterial cell and that this is the bacterial chromosome and this is the F factor. Majority of the case, this F factor exists as an independent autonomous cytoplasmic element separate from this bacterial chromosome. But in some instances, it integrates into this bacterial chromosome. Okay, that I am going to show bacterial cell. And this is the bacterial chromosome, and along with that the F factor also integrated. Such cells are called HFR cells. Such cells are called HFR cells. And where that insertion of that F factor happens in some specific sequence in the bacterial chromosome, and those sequences are called insertion sequences. As this F factor can integrate to the main bacterial chromosome, we can call the F factor as episomes also. Okay, so because that F factor possesses that integration property, we can call it as an episome also. And also in some condition, this F factor, integrated F factor, again will move out of this bacterial chromosome from the place where it inserted. And again, that cell get reverted to a F plus cell when the integrated F factor again leave the bacterial chromosome and start to remain separate as an independent cytoplasmic element. Then that F cell, that bacterial cell again become F plus cell. Okay, then some other points also we need to know, that is that HFR cells when it share that its DNA with another F minus cell that I am going to draw, F minus cell. This is an HFR cell. HFR cell, and it is going to transfer its genetic material to another F minus cell. F minus cell. Another F minus cell. It is having its own genetic material. Conjugation tube is forming, and an incision happens here, and the 5' end first directed through this that conjugation tube to other bacterial cell in this way. So that one strand begin to migrate to the other bacterial cell, and it reaches here. Okay, it reaches here. Then it acts as a template for the generation of the other strand, and in rolling circular method in unidirectionally, the other strand also forming. Okay, in such type of that conjugation from the HFR cell to F minus cell, that time it got a complete portion of this HFR cells genetic content, one set of genetic content. Such type of transfer is called complete transfer. And in some instance, as this incision happen and the one strand start to migrate to the next bacterial cell, before it completes the complete migration, some breakage happens in between that results in the incomplete transfer of that HFR cells chromatin content to this bacterial cell. That time, that transfer is called incomplete transfer. Okay, both conditions I think you understand. Complete what? Incomplete what? And again I am returning back to that complete transfer condition. When a bacterial cell got a complete transfer of the HFR chromatin, this cell also become another HFR cell, and also as it got two set a pair of chromosome, these are the ways called a diploid diploid zygote. And what about the other condition in incomplete transfer? Incomplete transfer means that genetic material of HFR cell is not completely migrated towards the other cell. That time also this bacterial cell have another name, that is called merozygote. Merozygote means zygote is having only a partial or half portion of the genetic material of HFR cell. Then that bacterial cell is called a merozygote. Okay, when the genetic material is received only partially, then that time is called a merozygote. And most oftenly, that merozygote or that partially transferred genetic material is destroyed by the enzymes that present in this bacterial cell. Okay. And one more thing, if the transfer of the HFR genetic material doesn't occur completely, then that bacterial cell will not change to either HFR or F plus strain. Instead, it stays as F minus strain itself. Okay, clear? Next, I am going to show you the image of HFR transfer, HFR genetic material transfer. In this picture, we can see a F plus cell with the F plasmid and the bacterial chromosome. And that F plus cell start to integrate to this bacterial chromosome, and that cell get converted to HFR cell. And this HFR cell, as it start to make association with another bacterial cell, that is F minus cell, through a conjugation tube is developing here. Incision is happening here, and through the conjugation tube, one strand is transferring to the F minus bacterial cell. And in this picture, we can see that the bacterial cell receive a complete part of the complete one strand of HFR chromosome. And after this, two conditions are possible. One condition is that migrated HFR chromosome can integrate into that bacterial chromosome and can turn that F minus bacterial cell to HFR cell, to its HFR cell. That is one condition. Other condition is that as the migration or the transfer doesn't occurs completely, only a portion of HFR chromosome enters the F minus cell, and that F minus cell stay as F minus itself. In the meantime, the partially transferred DNA portion is digested or destructed by the enzyme present in this F minus cell. Okay, I hope that is clear to you. By this, we finish today's class and section. Thank you. I'm an icon, man. I'm a girl about a 10. I should fuck her in a Benz.